Imagine a world where chemists can build molecules with the precision of a master sculptor, carefully arranging atoms in three-dimensional space. This meticulous process is akin to crafting a delicate piece of art, where every atom must be placed with exacting precision to achieve the desired structure and function. This is the power of enantioselective synthesis, a revolutionary approach that has transformed the landscape of chemistry. By enabling the creation of molecules in their specific three-dimensional forms, enantioselective synthesis has paved the way for advancements that were once thought impossible. Just as our left and right hands are mirror images of each other, molecules can exist in two forms that are non-superimposable mirror images, known as enantiomers. These enantiomers can have vastly different effects in biological systems, making the ability to control their formation incredibly important. Enantioselective cross-couplings in particular have emerged as a game-changer in this realm. These reactions are not just a technical achievement, they represent a fundamental shift in how chemists approach the synthesis of complex molecules. These reactions allow chemists to forge carbon-carbon bonds, the backbone of organic molecules, with exquisite control over the three-dimensional arrangement of atoms. This level of control is crucial for the development of new drugs, materials, and other advanced technologies. This ability to selectively create one enantiomer over the other has profound implications for a wide range of fields, from pharmaceuticals to materials science. In the pharmaceutical industry, for example, the efficacy and safety of a drug can depend on its chirality. The development of enantioselective cross-couplings is a testament to the ingenuity of chemists, who have harnessed the power of metal-catalysts and chiral ligands to achieve unprecedented levels of selectivity. These catalysts and ligands are the tools that make it possible to guide the formation of one enantiomer over the other. These reactions have not only revolutionized the synthesis of complex molecules, but have also opened up new avenues for drug discovery, materials design, and beyond. The ability to create molecules with such precision has far-reaching implications for many scientific and industrial fields. In the next section, we'll delve into the profound importance of chirality in the molecular world and its impact on various aspects of our lives. From the way drugs interact with our bodies to the development of new materials with unique properties, chirality plays a crucial role in shaping the world around us. Chirality, derived from the Greek word chir for hand, is a fundamental property of molecules that has profound consequences for their interactions with living organisms and the environment. This concept is akin to how our left and right hands are mirror images but cannot be superimposed on one another. In the world of chemistry, this means that two molecules can be identical in composition, yet differ in their spatial arrangement, leading to vastly different effects in biological systems. Enantiomers, though seemingly identical in their chemical formulas, can exhibit drastically different biological activities. These mirror image molecules can interact with biological systems in unique ways, sometimes with beneficial outcomes, and other times with harmful consequences. The subtle differences in their three-dimensional structures can determine how they bind to enzymes, receptors, and other molecular targets within the body. One striking example is the drug thalidomide, prescribed in the 1950s to alleviate morning sickness in pregnant women. This drug, however, became infamous for its tragic consequences. While one enantiomer of thalidomide had the desired therapeutic effect, the other enantiomer tragically caused severe birth defects. This disaster underscored the critical importance of understanding and controlling chirality in drug development. While one enantiomer of thalidomide had the desired therapeutic effect, the other enantiomer tragically caused severe birth defects. This case highlighted the necessity for rigorous testing and the development of methods to selectively synthesize and isolate single enantiomers. The thalidomide tragedy served as a wake-up call to the scientific community, emphasizing the need for greater precision in the design and production of pharmaceuticals. This devastating case highlighted the critical importance of chirality in drug development and spurred the search for methods to selectively synthesize single enantiomers. Researchers began to develop new techniques and technologies to ensure that only the desired enantiomer was produced, minimizing the risk of adverse effects. This led to significant advancements in the field of stereochemistry and the development of more sophisticated analytical tools. The difference in biological activity between enantiomers arises from their interactions with chiral molecules within living systems. These interactions are highly specific, much like a key fitting into a lock. The three-dimensional shape of the molecule determines how it will interact with other chiral entities, such as proteins and enzymes, which are themselves chiral. This specificity is crucial for the proper functioning of biological processes. Enzymes, the catalysts of life, are themselves chiral and often exhibit exquisite selectivity for one enantiomer over the other. This selectivity is essential for the regulation of metabolic pathways and the maintenance of homeostasis within living organisms. The ability of enzymes to distinguish between enantiomers ensures that biochemical reactions proceed with high efficiency and fidelity. Just as a right-handed glove fits snugly on the right hand, but not the left, one enantiomer of a drug may bind perfectly to its target receptor, while the other enantiomer may not bind at all, or may even produce harmful side effects. This analogy illustrates the importance of chirality in drug design, where the goal is to create molecules that fit precisely into their intended targets, maximizing therapeutic benefits while minimizing risks. The ability to synthesize and separate enantiomers is therefore crucial for developing safe and effective drugs, as well as for understanding the intricate workings of biological systems. Techniques such as chromatography and crystallization have been developed to achieve this separation, allowing chemists to isolate pure enantiomers for further study and application. Enantioselective cross-couplings have emerged as a powerful tool for achieving this goal, enabling chemists to create chiral molecules with remarkable precision. These reactions allow for the selective formation of one enantiomer over the other, providing a reliable method for the synthesis of complex chiral compounds. The continued development of enantioselective techniques promises to advance our ability to design and produce safer, more effective pharmaceuticals. The foundation for enantioselective cross-couplings was laid by the pioneering work of three brilliant chemists, Richard Heck, Eiichi Negishi, and Akira Suzuki. Their contributions have had a profound impact on the field of organic chemistry, providing tools that have become indispensable in both academic research and industrial applications. These chemists, through their dedication and innovative thinking, have opened up new avenues for the synthesis of complex molecules, which were previously thought to be challenging or even impossible to create. Their groundbreaking discoveries, collectively known as the Heck, Negishi, and Suzuki reactions, revolutionized the field of organic synthesis and earned them the 2010 Nobel Prize in Chemistry. This prestigious recognition not only highlighted the significance of their work, but also inspired a new generation of chemists to explore and expand upon their methodologies. The Nobel Prize announcement was a momentous occasion, celebrated by the scientific community worldwide, acknowledging the transformative nature of their contributions. These reactions, each employing a different metal catalyst, palladium for Heck and Negishi, and palladium or nickel for Suzuki, share a common theme: the formation of a new carbon-carbon bond between two organic fragments. This fundamental process is crucial in the construction of complex organic molecules, enabling chemists to build intricate structures with precision. The use of these metal catalysts has streamlined many synthetic routes, making them more efficient and accessible. This ability to forge such bonds with remarkable efficiency and versatility opened up countless possibilities for synthesizing complex molecules, including natural products, pharmaceuticals, and advanced materials. The impact of these reactions extends far beyond the laboratory, influencing the development of new drugs, materials, and technologies that have a direct impact on our daily lives. The versatility of these reactions has made them a cornerstone in the toolkit of synthetic chemists. The Heck reaction, for example, has found widespread use in the synthesis of pharmaceuticals, agrochemicals, and fine chemicals. Its ability to form carbon-carbon bonds in a straightforward manner has made it a go-to method for constructing complex molecular frameworks. This reaction has been instrumental in the development of numerous drugs and agricultural products, showcasing its practical utility in real-world applications. The Negishi reaction, known for its high functional group tolerance, is particularly valuable for constructing complex molecules with multiple reactive sites. This reaction's ability to accommodate a wide range of functional groups without compromising the integrity of the molecule makes it a powerful tool in the synthesis of intricate compounds. Its versatility has made it a favorite among chemists working on complex synthetic challenges. The Suzuki reaction, perhaps the most versatile of the three, is widely employed in both academic and industrial settings for the synthesis of a vast array of organic compounds. Its robustness and reliability have made it a staple in the synthesis of pharmaceuticals, agrochemicals, and materials science. The Suzuki reaction's ability to form carbon-carbon bonds under mild conditions has broadened its applicability, making it a preferred choice for many synthetic chemists. These reactions, however, initially lacked the ability to control the stereochemistry of the products. This limitation meant that the reactions often produced mixtures of enantiomers, which could complicate the purification process and limit their utility in applications where a single enantiomer was desired. The challenge of controlling stereochemistry has been a significant focus of research, leading to the development of new methods and catalysts that address this issue. In other words, they typically yielded a mixture of both enantiomers, limiting their utility in applications where a single enantiomer was desired. This challenge has spurred further innovation in the field, with researchers developing new strategies to achieve enantioselective cross-couplings. These advancements have expanded the scope and utility of the Heck, Negishi, and Suzuki reactions, making them even more valuable tools in the synthesis of complex molecules. The breakthrough in enantioselective cross-couplings came with the development of chiral ligands, molecules that bind to the metal catalyst and influence its reactivity, effectively steering the reaction towards the formation of one enantiomer over the other. One of the most iconic chiral ligands is BINAP, 2,2-bis-diphenylphosphano, 1,1-binaphthyl, a remarkable molecule with a twisted propeller-like shape. Developed by Ryoji Noyori, a co-recipient of the 2001 Nobel Prize in Chemistry, BINAP and its derivatives have proven to be highly effective ligands for a wide range of enantioselective reactions, including cross-couplings. The success of BINAP lies in its ability to create a chiral environment around the metal catalyst, influencing the approach and binding of the reacting partners. This chiral induction allows the catalyst to discriminate between the two possible transition states, leading to the two enantiomers, favoring the formation of one over the other. Since the advent of BINAP, a vast library of chiral ligands has been developed, each with its unique properties and applications. Chemists now have a diverse toolkit of chiral ligands at their disposal, enabling them to fine-tune the selectivity and efficiency of enantioselective cross-couplings for a wide range of substrates and chemical transformations.